Hello, my name is Helen Berman, and I'm a professor at Rutgers University, and I'm the former head of the RCSB PDB. And today what I'm going to be talking about is how we anticipate innovations in structural biology, since the PDB is the archive for all structures that have been determined. So in the next slide, I show you examples of the kinds of structures that are in the PDB. And this beautiful slide was done by David Gutzel, who works part-time for the PDB and at the Scripps Research Institute. And David organized this to show all the different types of macromolecules in the PDB organized by function. And you can see there is quite a variety. And the Protein Data Bank archives all of this information. And right now there are 129,000 structures of nucleic acids, proteins, and large macromolecular machines. The data are collected, stored, and globally distributed using modern methods, and this necessarily changes with time since we have to keep up with the different methodologies. The communities that use the PDB consist of industrial and academic scientists, which include structural biologists, chemists, computational biologists, biophysicists, biochemists, geneticists, and so on. And in addition, we have educators, students, and the general public. So it's a very large user base, which we'll talk about a little bit later. The PDB has grown over the years and continues to grow. The number of X-ray entries are shown, number of NMR. The growth in NMR is not quite as big as the growth in X-ray. The area that has really taken off and is probably the leader in terms of the new structural biology is electron microscopy. So you can see in the lower right the released entries as well as what has come in over the years, and things just continue to grow. And if you actually think in terms of molecular weight, the growth is really high because the molecular weight of the structures that are coming in continues to grow, which I'll talk about at the end of my talk. So what I wanted to do first is give you a little bit of history to see how this all came about and how the PDB came about and how we've been able to anticipate the changes, both community changes, scientific changes, and technological changes, so that we can keep on top of what's going on. So the early days of structural biology began in the 1930s when J.D. Bernal, with his student Dorothy Hodgkin, first crystallized an enzyme, and that was and took X-ray diffraction data. So that was a major event, and it was something that ushered in this relatively new science. Then Kendrew and Perutz determined the structures first of myoglobin and then of hemoglobin. And they were, again, these are the pioneers in structural biology, and these were Cambridge crystallographers. And I show in the lower right is a picture of Perutz on the left and Kendrew. You can see that there was a postage stamp made of Dorothy Hodgkin, who was another pioneer in this field. So these structures began to be determined, and the first one, myoglobin, was determined in 1957. And a few structures, one at a time, began to be determined. And some people in the scientific community felt that there was real important information in these structures, and that there needed to be a way that the structural information could be shared. So remember, this now was in the 60s and very early days in terms of networking and computing. Nonetheless, it was felt that some kind of an archive should be established. So in 1971, well, actually started in 1966, but by 1971, there had already been a petition in which people asked that there be some kind of an archive. I have to tell you, I was part of that petition, and I have absolutely no idea who we thought should receive the petition. We just did it. And then in 1971, there was a meeting at Cold Spring Harbor in protein crystallography, and it was organized by Jim Watson. And basically all the famous crystallographers came, and you look at the upper picture is Hal Wyckoff on the right, who was one of the people who determined the structure of ribonuclease. And importantly, at that meeting was Walter Hamilton, who was a small molecule crystallographer from Brookhaven National Laboratory. And Walter basically crashed the meeting, drove down the Long Island Expressway, came to the meeting, and a whole bunch of us met him in the bar at the end of one of the days. And we said, we really need to have an archive for these structures. And again, there were very, very few. And he said, okay, done. And that's how the PDB began. And on the right, so the next thing that Walter did was fly over to England, where he met with Olga Kennard, who had a small molecule database, and they agreed to have a collaborative enterprise between England and America to collect and distribute these data. And Walter understood the importance of an international collaboration. And so the PDB began. On the lower picture shows you another picture of Walter on the left, and on the right is a picture of, to the right of me, who's also in this picture, is Tom Ketzel, who took over the PDB after the very untimely and early death of Walter. So that's how the PDB began. At the time when we started, there was probably about 7 structures, and everything was done on punched cards. Tom in particular wrote letters to every protein crystallographer asking them to please submit their data to the PDB. And then there was one curator, Francis Bernstein, who curated the few data sets that existed at that time. So this went on for many years, there in 1982, when the AIDS epidemic began to be apparent, the NIH was very concerned that the data, the structural biology data that was relevant to HIV would not, they did not want it to be in any way held back, and they wanted it to all be public since this was a public health crisis. There were also people like Dick Dickerson who felt very strongly that the data had to be made public, and that article is called The Missing Crystallography Data. He made quite a big fuss. Some of us wrote letters, like myself, to various journal editors asking that there be a requirement for data deposition. Fred Richards from Yale led a campaign to insist that these structures be put into the PDB. So everyone, there was a feeling that everything had to be in the PDB for the public health and for the public good. So in order for this to happen, committees were established of crystallographers to talk about what data are we talking about anyhow. What kind of data should be in the PDB? When should they be submitted? How could they be retained, for how long, and so on. So there were about 4 or 5 committees that were set up. I show some of them here to figure out exactly how to do this. This process began in about 1982 and concluded in 1989 when guidelines were set up and published by the International Union of Crystallography, which stated exactly what data had to be in the PDB and when they had to be put in the PDB. So this was all a community effort, which was actually quite successful. Once this happened, then the NIH, the NIGMS set a requirement that structures had to be in the PDB. So now we have the requirement for structures in the PDB. The next thing is to get a little, if you're going to make a database, they really need to be, it needs to be searchable, and I'll come to that later. In order for it to be searchable, you have to have some kind of standards for how you do this. So in the early 1990s, the International Union of Crystallography set up a committee to create standards for crystallographic data, and that was called the Macromolecular Information Format, MMSIF. The first draft of that was in 1996. The people involved are shown on the upper right as John Westbrook, Phil Bourne, who you might recognize, Paula Fitzgerald, who at the time was at Merck, Sid Hall, who was the author of the small molecule SIF, and Shoshana Vodak were among the people on the working group, as was I on that working group. So we got this dictionary made that contained all the terms. It was about 3,000 terms that would describe the crystallographic experiment and the results. This dictionary later became the underpinnings of the RCSB PDB, but it was not a widely accepted dictionary or standard. As the number of structures began to increase, it became apparent that we needed to do something to have enable a round trip of the data that comes out of refinement programs, goes into the PDB, comes out of the PDB, goes back into refinement programs, into structured determination packages. And the way things were, a lot of people spent a lot of time doing format conversions, and it was really a waste. So we had a meeting in 2011 where it basically the PDB group and the software developers came together, and basically the PDB said, we'll take any format that you want as long as you all agree on a format. And originally there was some talk about some other kind of format, but then on the second day of the meeting, the software developers said, this is kind of silly, why don't we just use MMSIF? So this was decided in 2011, many years after the creation of SIF. There were already definitions for X-ray, and the PDB had definitions for NMR and electron microscopy. And so and because it's no longer just crystallography, it was called PDBX, and that was the master format, and that continues to be the master format for the PDB. If you remember, I said that when the PDB was established by Walter in 1971, he knew instinctively how important it was going to be that this whole enterprise be global. And so, but there was no formal plan for how to do that. So in 2003, we created a memorandum of understanding establishing the worldwide PDB. At that time, I was involved. Kim Henrik was at the PDBE in Hingston, and Haruki Nakamura in Osaka. And the agreement was that we would collaborate on all aspects of data processing and annotation, so that no matter where the data are handled, it'll always have the same format and the same data processing standards. We also agreed that each of the sites could provide different services and different websites for those kinds of data. And we have an advisory committee, which continues to be very, very active, an international committee consisting of leaders in X-ray, NMR, and EM. Okay, so the final piece of the puzzle in this case was that if you're going to then validate these data and make sure that the data that are in the PDB are correct and the coordinates fit the primary data, you really have to have the primary data to validate against. And again, there was lots of articles written and community involvement saying that structure factors needed to be in the PDB. And finally, in 2008, structure factor amplitudes, intensities are required in the PDB for crystal structures. And now we also have the requirement of restraints and chemical shifts for NMR. And very recently, the maps for EM are also required to go into EMDB, so that all of the data has a chance of being validated. So what I've just shown you is, so far, is that we had a lot of community involvement, and what I wanted to point out is that we had these task forces in each field which went through and very carefully decided how you should, in fact, validate structures determined by the different methods and come up with recommendations. Once they come up with recommendations, this is then implemented in the PDB. So the X-ray validation force recommendations are fully implemented. The NMR validation task force, most of the recommendations are implemented, but they're continued to make more. The EM validation task force continues to be very active in trying to come up with validation standards for EM. Recently, there are now recommendations coming from small angle scattering, which is a method used more and more in conjunction with the other methods for structure determination. And as I'll talk about a little bit later, we have the emergence of hybrid methods where we had to have a workshop in order to figure out how we were going to handle these kinds of structures, which I'll talk about at the end of my talk. Okay, so what I've done, I think, is tried to trace for you sort of the evolution of the PDB and how over time different things had to be considered. So first and foremost is the science. The different kinds of structures were determined at the beginning. Right now we're dealing, and I'll talk about that a little bit later, the large macromolecular machines. The technology for determining structures continues to change. In the old days, we were using punched cards, diffractometers, pretty clunky machines. In the 80s, the emergence of the use of synchrotron radiation enabled people to work with smaller and smaller crystals because of the high intensity beams. Computer graphics emerged as a very important part of structure determination and of course continues to be heavily used. In the 90s, we see the emergence of electron microscopy, very fast computers, very fast detectors. In the 2000s, we have the appearance of, through the Protein Structure Initiative, high throughput structural genomics and the use of robots so that structures now can be determined rapidly and actually much more precisely and accurately than in the old days. That was a very big result of that initiative. And now we're dealing with hybrid methods where structures are being determined by multiple methods, and we have to figure out exactly what to do. And of course, I've emphasized and have to continue to emphasize the importance of community. So in each of these different eras, different things were important. First, community involvement in getting the archive established at all. Then in trying to figure out what the guidelines for deposition should be, headed up by, very strongly influenced by Fred Richards. Then we had the standardization of coming up with standards for data representation. And then in 1998, the PDB moved from Brookhaven first to RCSB PDB, which was a consortium consisting of Rutgers, NIST, and UCSD and SDSC. As time went on, data centers were established at the EBI, PDBE, and Osaka, PDBJ. And so we formed the WWPDB. And then finally, the big push on figuring out what to do next, which absolutely requires community, is the creation of validation standards. So this is a complex operation. You need to think about all aspects in order to move forward, and you kind of have to be a little bit read the future, trying to figure out where the emphasis needs to be. Okay, so now we have a PDB. How exactly are the data processed? And how the PDB has a long history of evolution in the area of data processing. So the pipeline consists of first the creation of the data in the lab by the research scientists. Then once the data are created, it's deposited in the PDB. It's processed, which we're going to talk about how that's done. It's archived and it's distributed. And this pipeline is true for any data project. You have to consider all these aspects and how you're going to handle them. So in the early days, the data processing was done by a lot of standalone software in different groups. Before we became the WWPDB, the software was completely different in the different countries, and we had to align the results and make sure. So what that meant is that we had to move data back and forth between Europe and America to double check that the results would be the same no matter who did it. And that was a lot of effort. And so about 10 years ago, it was decided that we needed to have a single pipeline, and this would be a collaborative project by the WWPDB for deposition and annotation, and this was called the OneDep system. And it provides support for X-ray, NMR, and electron microscopy. The file formats are standardized, and we have controlled data dictionary, so we're all talking the same language. We're able to collect more data. We were facing the issue of very complex structures, very large structures, and we needed to be able to in the old PDB format, something like the ribosome was split into many files because of the restrictions of the format, and with the new systems, that is no longer the case. There's also improved efficiency through automation and validation, and then very importantly, we have workload balancing based on resource capacity and location, and that means that And these are real life things, as you will hear, that happen. If there's a flood in New Jersey, the people at the EBI can take over. If there's some kind of a computer meltdown at the EBI, then the people in Rutgers can take over. If there's a tsunami in Japan, we can all cover for one another. So the normal way is the data are distributed, are handled geographically, but because it's all one system, anybody can process anybody else's data, and that was really, really important. So the one-depth system, this is just another schematic of what the workflow is. We have a deposition pipeline where we have the deposition interface. We have the data upload and harvesting. There's reviews, there's validation, and then submission. And then once it's received by the PDB curation staff, there's very specific ligand processing, sequence processing, validation, other kinds of annotations which have evolved, and then finally release processing. So in annotation, we add metadata to put the data in context with the science. We aim for consistency, and we aim for standard vocabulary and terms. And that's taken some time to get to that place. Ligand annotation is actually much more difficult than the polymer annotation because every ligand is different. The chemistry is completely different, so it has to be reviewed very carefully. So we get all the author-provided chemical information, and we look at it in 2D and 3D. We also look at the electron density to make sure that the ligand fits in the map. And as you can see in the top right, this ligand fits very well, whereas in the bottom left, it doesn't fit so well. And so we have to check all of that, and that's done in an interactive way with the depositor. For sequence annotation, one of the biggest problems that used to happen in the PDB was that the biological sequence and the atomic coordinate sequence didn't match. Sometimes that's real because it really isn't the same, and sometimes it's a mistake, and there's all sorts of reasons for that mistake. And so we have a very clear way of checking that and looking for any kind of discrepancy, and then if there is a discrepancy, describe what that discrepancy is and why. Then, as I mentioned before, we have spent a lot of time on validation, and we now have a validation report. Everybody wants to have a single number that says how good a structure is. Well, that's just doesn't work. So we have a thing called a slider, which was recommended by the task force. And everything on the right, if it's on the right, it means the structure is okay. If it's on the left, there are some problems. And various aspects of the structure are summarized on this slider and help people decide whether or not a structure is okay. The other very important validation indicator is a residue plot. And so if something is green, it means it's all fine. If it's red, it's not so fine, and then everything in between is what it is. And so this structure is actually not too bad. The bars indicate the geometry aspects, and then on the top you see a little red dot. That little red dot means that the match to density is not good. So for those two residues in this case, there's some problem. So the depositor receives a very detailed report. They often go back and re-refine the structure when they see there's a problem. And then finally, when the whole thing is over, there is a PDF that is can be uploaded with a manuscript and submitted to a journal. And now a very impressive number of journals are requiring validation reports as part of the journal review, and that, of course, will make the quality of what goes into the journals get much better. So what about impact? If you're going to have a data resource, then you have to be able to show that it's useful, and you have to measure the impact. And if you don't have much of an impact, you have to then think about what you're doing. So impact is really an important characteristic of a data resource and needs to be studied. And so the PDB has been doing that for many years, and we have several indicators. So first of all, we have some numerical indicators. We have about 11,000 new structure depositions per year, so that's still pretty healthy. The biocuration responsibilities are distributed by geographic location, and that's shown in the middle. What's really impressive to me is 1.5 million coordinate data files are downloaded per day. So that's a huge number of data files that are being downloaded in a day. And the pharmaceutical companies use the PDB inside their firewalls daily. And they download every time there's an update every week, they download all the new structures and put it into their own databases. Another sign of the use of the PDB is our citations of the PDB, all different kinds of citations. When the RCSB PDB first took over the PDB in 1998, we published in the year 2000 a paper in the database section of NAR, and it just described what we were doing. And that paper now has been cited by more than 21,000 times. It's in the top 100 papers in science ever cited, so it has a lot. So then we say, well, who's citing it? Well, it turns out that it's not structural biologists who are citing it. It's people who are using the data for other kinds of analyses. And we've just completed, and it's not shown here, another analysis of this paper, trying to understand what they're using the data for. And it's not surprising that biochemistry and molecular biology are using the data, but computer science ranks right up there. And when we did in this analysis that we did, we discovered that there was a disproportionate amount of analysis and use of this paper by computer scientists and plant biologists. And now we're trying to understand what, in fact, they are doing with these data. We've read some of the papers, tried to understand what they're doing. So these are people who are doing analyses of structures in the PDB, sort of large analyses, and are citing this paper as their source. We also did an analysis of patents that mention Protein Data Bank, and there's a 3166, and a new analysis was just done where double that number are shown in patents. So that's another indication that the data are being used. Another analysis that we did is who uses the PDB, which other databases rely on the PDB, and we discovered by looking at the NAR journal that the PDB data is used by more than 200 biological databases. And since 2011, 25% of new databases utilize PDB data. So it's 119 out of 452 new databases utilize PDB data. So again, the PDB data are used a great deal by other people. So without knowing it, we conform to the FAIR data principles. Now, I can assure you, when we began this enterprise, FAIR had not been defined, but it's findability, accessibility, interoperability, and reusability are the definition of those guidelines, and it looks, the PDB seems to be following those guidelines, those principles very well. So what I've shown you until now, I hope, is that the PDB grew sort of organically. I grew with science that it was archiving, and it changed as the science changed and as the public awareness of the importance of data changed. And that's something that has to be done all the time, keeping your ear to the ground while you're trying to build something. And of course, the other thing that's very important, I think all of you would know this, is that we have to make some compromises. If we use bleeding edge software, we could get into trouble where things are not working. So we have to keep moving forward without breaking things. And so that's a challenge all the time, and testing and making sure things are working has to be done. And even with that, there's plenty of room for errors, and we certainly make them. But at least, you know, we're thinking about it all the time. Okay. So the next big challenge sort of crept up on us, and we have to face, and that is integrative structural biology, which I'll talk about a little bit. And in thinking about integrative structural biology, we have to see whether what we did before with the PDB can now be applied going forward. So what is integrative structural biology? This is from a paper by Ward and Shally, and what this field is that you have data from more than X-ray, NMR, and electron microscopy. Either any one of those methods on their own can define a structure, but there's also data from small angle scattering, cross-linking, HDX, MS, proteomics, co-purification, bioinformatics. All those different areas provide some kind of data. And so you get all these components in these large macromolecular machines that you want to get a structure for. And so how do you do that? And so you have to take the data, and you have to use computational methods, and eventually you get a model. And so this field has been developing over the last few years. And at the beginning, we really did not know what to do. People were determining these structures. So this is no longer crystal structures or small proteins in solution, or even large proteins that are done with electron microscopy. These are really large machines. So there were some attempts to deposit the structures into the PDB, and at the beginning, trying to be accommodating, we took them, but we realized we had no idea what to do with these structures, and we didn't really have mechanisms for processing these data, and certainly we had no way of validating these data. So what we did is we didn't want to lose the data for anybody, so we put them somewhere, so when we finally figured out what to do, they wouldn't be lost. And as the PDB has always done, we decided we have to reach out to the community again and find out what they want. And so what we did is we brought together people from the various fields involved. So that involved NMR, X-ray, EM, small angle scattering, computational modeling, and so on. So it was a pretty large group of people, and Harald Kleivik hosted the meeting at the EBI, and we all came, and Torsten Schwedt is in this picture, this Klaus Seidel, who does FRET, and we all came together and we figured out, you know, what should we be doing with these structures and how should they be handled. So these are the recommendations that we came up with. First of all, be as inclusive as possible in collecting data from the many experimental methods. So we have to allow for all the different methods. Accommodate many different types of structural representation. So what that means is, right now we're used to looking at ball and stick for smaller molecules, ribbons to represent ribbons and helices, which is the Jane Richardson representation. We have the space filling representation, but sometimes we don't have any idea. We know that the structure is in a sort of area and they're sort of blobby. So how do we represent those blobs? And that's a big decision we have to make. It was also recommended to create a federated system to collect and curate the data, which I'll show you in a moment. Use a common interface to collect the data. It was asked that the WWPDB play a leadership role and that we write a white paper. So all of that happened, and we did write the white paper to describe what it is we wanted to do. We then, once that white paper was out in May of 2015, we got to work and we made some subgroups. We had a federation subgroup, which is to create a network of model and data repositories that would interoperate in some way with the PDB. And Jill Tuella, who was from the University of Sydney and University of Utah, and myself head up that subgroup. Then there's a model validation subgroup headed up by Andre Schally and Torsten Schwedt to create standards for representation and validation of these models. And so these subgroups are very actively working, trying to figure out what to do. So give you a sense of what the puzzle looks like. There's data from all these different places. How do you put it all together? How do you get it so that the individual communities involved, for example, small angle scattering community, are able to put whatever data they want into a repository, but at the same time, the restraints that are generated from small angle scattering would be shared with the PDB for validation of these integrative models. So we have all these different, we have things that are established already, which are shown in color, and we have other things where we're working with the communities to help them set up their own data repositories. And then the model for the federation is shown here. So in the middle are the model repositories, the PDB, which is right now X-ray, NMR, and EM structures. There's the model archive, which is set up in Switzerland by Torsten, which has purely in silico models. And then for now, we have something called PDB Dev, which PDB Development, which is the place where we are working to put whatever hybrid models exist now and experiment with figuring out how exactly to validate these models and how to archive them to begin with, how to validate them. So that's in the middle. These are structural repositories, and our proposal is that the only thing that would be in there would be the coordinates and metadata, as well as the experimental data that's enough to validate the model. At the same time, on the outside part of this thing, we have the individual repositories for specialist fields, such as right now EMDB exists for cryo-EM maps. BMRB has all kinds of NMR data, only some of which is shared with the PDB. So we have models for how this works. SAS-PDB is established in Hamburg for small angle scattering. And then the other ones that we hope will come on board are shown in this. And so what would happen is that the different methodologies would be able to handle the data however they wish, and then share what is required for validation of models with the PDB. So in order to make this all happen, we need methods for data deposition and harvesting. There's a lot of data here that you can see. How exactly do we curate? How do we archive? Then how do we move the data back and forth between these small repositories and the PDB, and how do we distribute? So this is all something that has to be considered for this kind of federation. This is a proposed federation workflow. So we have the different kinds of modeling platforms. Right now there are a few. We have the PDB Dev, which is the repository that we're using as a test bed for this whole project. We have then the data dictionary, and we're working with each of the groups to come up with data dictionaries that would allow data to flow between their databases and the PDB Dev. And then we show here the model repositories at the bottom, the experimental data repositories, GitHub, which is used a lot, and the NIH Commons, which I hope will happen. And then there's also unhosted data sets that are just out there, and we have to figure out how to bring this all together. We're going to do this by building on the OneDep tool, which already has X-ray, NMR, and EM, and we want to figure out how to add the other methods, but this is a framework that will allow us to think about this in a more reasonable way. We've done a huge amount of work on the dictionary that underlies all this, and we have terms. We have terms for the molecular components, the structural models that are, and the data from the structural repositories, and then data from the experimental data repositories, and we have terms for all of the everything that's involved in those repositories, and then all of that goes into the integrative modeling, and on the other side you get the ensembles of the macromolecular assemblies. So getting this dictionary put together and being able to do this will enable the kinds of data exchange that's going to be required for this project. As a test, we tried to see if we could use the test dictionary, the pilot that we developed, to represent a multi-scale model of the nuclear pore subcomplex. And you can see that this was a completely dictionary compliant, and we visualized it using Chimera. So in our first experiments, things are working quite well. So what I've talked about with you are the many different facets of building structural archives. The PDB, we have the RCSB PDB, PDBE, PDBJ, and BMRB. We had a celebration when there were 100,000 structures in the PDB. That was a big deal. Now we have 130,000. On the right, upper right, is a picture of a celebration of the 40th anniversary of the PDB, and we quite appropriately held that meeting at Cold Spring Harbor, which we view as the birthplace, because it was there that Walter Hamilton agreed that there should be a PDB in 1971. And then, of course, there's the funding that we've gotten from NSF, NIH, and DOE in America, and then EMBL, Wellcome Trust, BBSRC in Europe, and then funding from Japan, and funding for BMRB and NIGMS. So there's a lot of different grants that get pulled together in order to keep this thing going. The IHM project is just at the beginning. Right now, it's a collaboration between Andre Shally at UCSF and my group at Rutgers. We have these subgroups that I told you about, and the names of the people on the subgroups are shown in the slide, and Jill is heading up the Federation one, and Andre and Torsten the model validation. Right now, the funding that we have had has been from an NSF EGA grant, which allowed us to build that first pilot dictionary, and we're currently seeking funds to continue building what we think is going to be a very important part of the PDB. And I guess my goal in 5 years from now would be that all of these kinds of structures would be able to be in the PDB, fully validated in ways that you could understand how good or bad they actually are. So that's what I have to say. I would be very pleased to answer any questions if you have them.
Okay, well, thank you everyone for tuning in, and hopefully you'll join us for our next webinar.